Hello and welcome in my Biosensors course. This is your instructor, Tanu Bharadwaj. I have spent more than 10 years in this field. I am excited to share my knowledge with you. Now we are going to take a dive into the fascinating world of biosensors, and you are going to learn how to design your own biosensor. This course contains practical activities, quizzes, projects, and downloadable resources. I am thrilled to have you here. Let's dive in and start building your biosensor today. Feel free to post your questions. I am here to support your learning journey. So, happy journey and let's start the course. These are the content of this course. We are going to learn about biosensors, different components of a biosensor, why biosensors are important, the basic terms to define biosensors performance, different types of biomolecules, transducers, and immobilization techniques. After all this, we are going to study about types of biosensors with an example of an aptamer based biosensor, various applications of biosensors, then how to select right components to design a biosensor according to the application. Lastly, different examples of biosensors so that you can design your own biosensor. Before starting with the topic biosensors, I want you to have knowledge of this term, interdisciplinary area. So, what is an interdisciplinary area? This is an area of study that involves knowledge of different fields or disciplines to address complex problems or questions. Benefit of this field is that you can look into the problem from different perspectives or dimensions, resulting in solution of the problem, easy solution for the problem. Biosensors is a perfect example of an interdisciplinary area. This can involve integration of various different fields. For example, fundamental sciences, physics, chemistry, biology, engineering, biochemical, chemical, or material science. This can also involve cutting edge techniques like nanomaterials, microfluidics, bioinformatics, etc. So, now the question is, what is a biosensor? Biosensor was invented by Clark in 1962, and according to the definition, biosensor is self-dependent bioanalytical device which has biomolecules layer in intimate contact with the transducer, resulting in electrical signal. So let's see how a biosensor work using a block diagram. The main part of a biosensor is a transducer, which is present in intimate contact with a layer of a biomolecule. The biomolecule can be of different types, which will be discussed in the coming lectures. So, after arrival of the analyte will bind to the biomolecule and they will all together generate a biochemical signal or biochemical change. Now, this signal will be sensed or detected by the transducer and will be converted into an electrical signal. This electrical signal will further be amplified and processed by the electronic circuit, and it will be displayed on a display unit. So these biosensors act as bridges between the biological world and the electronic world, allowing us to detect and quantify a vast range of substances with increasing accuracy and speed. Now that concludes our quick introduction to the interdisciplinary area of biosensors. Thank you. Let's meet in the next lecture.